Streptococcus agalactiae was the main in the 85 of the animals. That was Strep agalactiae alone. We also have others, but now the core occurrence was mainly Strep and Strep uberis, Strep agalactiae and uberis. That was only 4 farms, and then you can see the others distribution there on the upset plot. The final objective was to assess AST profile. The main thing that you can see here is just that tetracycline, all the isolate, the strep isolate, were resistant to tetracycline. For the gram-negative now, these were the panel of antibiotics that were tested against, and the main thing that we can see there is the trimethoprim, and then there's tetracycline again at 33.3%. So the conclusion is mastitis remains high prevalent and is influenced by both animal factors and farm level factors, as I've just said. That is including the parity, the use of dry cow therapy, etc. So when we want to address the issue of mastitis, we need to integrate intervention combining farm training, improved biosecurity, hygiene practices, and we strengthen the veterinary farm engagement, and also the issue of antimicrobial stewardship strategies. Now, I like to acknowledge these people, or these groups, the dairy cooperative, because that's where we got the sampling from, and also they provide the logistic support. The county government and the national government, because they provide the approval, and also NACOSTI. And farmers, because they allowed us to visit their farms and also provided the information that we are presenting here. Also, we have the funders. And this team, that's the team that conducted the work, including me. I'm not there, but thank you.
Thank you so much to Eugene. Let's give him another round of applause. He's actually saved us 3 minutes. So keep noting down your questions. We'll have a QA session at the end of the presentation. So just to confirm, Teresia Nyaga is in the room. George Gitau is in the room. And Sarah, I think I've seen her. So one of the take-home messages from what Eugene has presented is that indeed addressing mastitis will require an integrated intervention, including AMS training, and that's one of the reasons why we are here today. So our next presenter will be Teresia Nyaga. Teresia, you can just come up as I load your presentation. And Teresia will be presenting on, oh sorry, this is, and Teresia will be presenting on antimicrobial susceptibility profile of E. coli isolates received in National Microbiology Reference Laboratories for retesting period of 2022 to 2023. Welcome, Teresia.
Thank you so much. Good morning, everyone. Good morning once more. Yes, I'm excited to be here on this week and to present. This is our work. The work has been done at the National Microbiology Reference Lab. So this abstract has been a collaborative idea, and you can see there are several co-authors in the list from various institutions, from NPHI, NACI, KICAP, Fleming Fund, and University of Nairobi. So this is how it's going to be. We'll have a background, look at the methodology, the objectives, then we'll have the results and the conclusions, stroke recommendations, and the acknowledgements. So, as we have heard from since yesterday, antimicrobial resistance remains a global challenge. And in 2019, it was attributed to 1.3 deaths. And then it was associated with 4.7 deaths. And actually, according to a report that was released this year, in every 1 in 6 infections that are caused by bacteria, there is an antibiotic-resistant microorganism. So it should really concern us. And when we have antimicrobial resistance, what happens? We have increased healthcare costs. The patient's outcome tend to be severe, of course, with increased morbidity and mortality. The treatment options are limited because then the doctors or the clinicians will have to garble between limited antibiotic choices. Probably this patient has complications, so you can imagine the dilemma that it puts the clinician in. Then there's also prolonged hospital stay. And of course, this will also expose the patient to even chances of acquiring hospital-acquired infections. So why are we looking at Escherichia coli? Number one, it is a priority pathogen for AMR surveillance. It is also a One Health indicator organism, and we know it is associated with community and hospital-acquired infections. And looking at the infections that are attributable to E. coli, in 2022, E. coli was associated with 250 million infections and 65,000 deaths annually. That was a study done in Cambridge. And certain strains have also developed antimicrobial resistance, like they have been able to gain the resistant genes. So the overall objective was to determine the antimicrobial susceptibility profile of Escherichia coli isolated from national microbiology reference isolates that were referred for testing between 2022 and 2023. And this is the methodology we used. So we receive isolates from Sentinel AMR sites, and when they come to the lab, we re-identify these microorganisms using MALDI-TOF. And then, based on the organism that we get, we judged based on the national guidelines, which include the surveillance strategy, the CLSI, there is a Kenya Clinician Handbook, to determine whether that pathogen is, whether that bacteria is a pathogen in that site where it is being isolated. So those that are identified as pathogens at the site of isolation, we proceed to do antimicrobial susceptibility testing. Mostly we utilize the Vitek 2 compact equipment, and then we archive them, of course, for future studies. And from there, that's where we analyze data for E. coli using Microsoft Excel and WUNET software. So these are the results. So for that period of time, we had 1931 isolates, out of which 653 were Escherichia coli, and these were coming from 12 AMR Sentinel sites. From the graph, I'm not sure whether you can see it clearly. You can see urine had the highest numbers, and tissue and the least numbers, and of course there are other specimens that were diverse. This is the susceptibility profile. So looking keenly, I'm just trying to compare this with what Eugene has presented. He didn't have ampicillin, but you can appreciate that ampicillin is doing pretty poorly at 88% resistance. Yeah, and then cotrimoxazole, just like the previous presenter I had, again, it has quite high resistance. This is in human sector. Remember, that was in the animal sector. And then cefuroxime followed closely. And in terms of the susceptibility, nitrofurantoin, of course, this is in urine samples, was still working very well. Amikacin, we have meropenem there, the gentamicin, the piperazoline, tazobactam, doing pretty well. So in terms of the resistance to more than 3 antibiotics, which we call multi-drug resistance, from the isolates we had for that period, we can see 28% were MDRs. Sorry, we didn't look at the XDRs and PDRs. And you can see, of course, the wood isolates and the highest number of the MDR Escherichia coli. So what are we concluding? There are alarming levels of antimicrobial resistance in clinical isolates, and there is a significant number that are MDR multi-drug resistance. So we recommend, as the week slogan goes, we act now, we protect our present and secure our future. We need to act now on our AMR surveillance sites, rather activities. We ensure we are monitoring the surveillance patterns so that we don't continue prescribing the antibiotics that have already gained resistance. We also recommending judicious use of antimicrobials that are available. We strengthen our antimicrobial stewardship committees. We ensure antibiotics are not used where there are no clinical indications. And again, we encourage completing the dosages that are recommended. And then we also we are recommending infection prevention measures to be put in place, and we all adhere to them because this will reduce our need to use the antimicrobials. We also recommending adoption of new therapeutic strategies. Things like phage therapy could help where antimicrobials are not working. And as I said, these isolates have been biobanked. We only know the phenotypic characteristics. We'd also want to know which resistant genes are contributing to these phenotypes are doing. Could there be other resistant genes that are coming up that we could pick up through molecular characterization? So I wish to acknowledge the Ministry of Health, the NPHI where we work, the Fleming Fund have been working with them, and yes, the ECAP Kenya, they supported me in this work. Of course, I work with Fleming and the University of Nairobi, the Infection Prevention Network. I think they first taught me how to write abstracts, and the Association of Public Health Laboratories. And as I conclude, I reiterate this week's slogan: we act now, we protect our present and secure our future. Thank you very much.
Okay, let's appreciate her once more. Thank you for that insightful presentation. So our first two presenters have told us about how E. coli is showing resistance. And Teresia has mentioned that E. coli is a One Health indicator organism. So E. coli can be found in humans, it can be found in animals. And yesterday, one of the presenters from Water Resources Authority mentioned that they're doing some surveillance in the environment. And one of the organisms that are being targeted is, of course, E. coli. So bringing it back home, we should really protect our antibiotics and use them prudently because, as Teresia has mentioned, 1 in every 6 infections is caused by resistant bacteria. That should be very worrying because it can be you, it can be your family member, it can be the animals that you're keeping at home. So let's work to protect our antibiotics and antimicrobials in general. So for those who have come in late, today we are celebrating International Men's Day. So again, turn to the man next to you or behind you, tell them Happy International Men's Day. And make sure to post a man in your life. And as you post them on your social media, don't forget to also post about antibiotics. Is that right? So, as you post, also don't forget to post about the WOW week and tell people to use antibiotics prudently. Okay, so I welcome the next presenter, George Gitau, who is going to tell us about modeling antibiotic resistance in WHO priority pathogens in Kenya, a machine learning and epidemiological approach. Welcome, George.
Thank you very much. I'm really privileged to be here today. I'm George Gitau Wanjera, and I'm currently finalizing my MPhil at the University of Cambridge, where I pursue an MPhil in population health sciences, and I focus on infectious disease epidemiology. And today I will be sharing this work that I did in collaboration with a friend of mine who just walked in, who's a machine learning engineer, as part of the Vivli AMR Challenge 2025. So, the title is Modeling Antibiotic Resistance in WHO Priority Pathogens in Kenya, a Machine Learning and Epidemiological Approach. So, we know the problem, right? I mean, that's why we are all here, Wow Week. So, we all know that AMR is a severe global health threat that we are currently facing, and that is projected to get worse if don't do something. And a common challenge in our setting is that by the time we get the full microbiological report, the patient may have already been on ineffective therapy for days. So our work seeks to ask, what if we could flag a high risk of MDR from the moment the sample is taken and from the moment the initial tests are undertaken? And so we set out to conduct this study, and our main objectives, our study objectives, were to estimate the AMR and MDR trends, to develop predictive machine learning models, to generate explainable insights, because we do not just want to give a black box of a model that cannot be interpreted easily by the clinicians without, again, adding to their burden. And so we used sharp values. I will explain that a bit later. And we also wanted to demonstrate a prototype clinical risk flagging tool for early MDR detection, and hopefully translate these outputs into stewardship and surveillance recommendations. So this was our data set, the Pfizer Atlas Kenya data set from 2013 to 2023. And this data set has its strengths and limitations. It is longitudinal, it is structured, and it is standardized. However, we quickly realized that there were some limitations, such as missingness, no clinical outcomes, and while we had initially planned to work with about 20 years, we had to work with 10 years since that was what was available. It also does not categorize our data by regions, so we had to work on Kenya as a whole, and we could not conduct any geostatistical tests. But it also has some good features, such as patient demographics, specimen type, and antibiotic susceptibility, resistance susceptibility, So, from the data to our prediction, so what was our machine learning approach? So we cleaned and imputed our data using mice, and we also defined MDR as resistance to more than three drug classes. We wanted a model that could find the hidden patterns, but we knew that we needed to first of all have a baseline. So we began with a logistic regression, which is simple, interpretable, and strong for baseline comparisons. But then we conducted a comparative analysis of different machine learning approaches and found that the XGBoost model was the most powerful in terms of the outcomes, which are in terms of its features, which I will describe in a moment. And our justification for using this was that it handles nonlinearities in the data, it's robust to missingness, and has a strong performance with tabular clinical data. And we, as I said, we prioritized sharp explainability to answer why. And so sharp basically tells us how much did each feature contribute to this specific prediction. I will show you an output of that in a moment. So this is the clinical usefulness of the model, in short, the model performance. And so model evaluations, we conducted an 80-20 train test split and 5-fold cross-validation. And our metrics that we
AMR prediction has actually turned out to be a great tool. So I'm privileged to introduce our last presenter, who is Sarah. So Dr. Sarah Kindiki, please come forward. You will be presenting on genomic analysis of Pseudomonas aeruginosa recovered from surgical site infections from a referral hospital in Western Kenya, reveals dominance of high-risk subtypes. Welcome.
Thank you so much. I'm going to present some work done at Kakamega County, and it's about the PA, which actually has revealed some dominance or has revealed some high-risk subtypes. This was from surgical site infections of patients who are attending the hospital, our major hospital in Kakamega. And as we know that surgical site infections are actually, they occur in areas where the surgery is made, and it's a significant global concern. And approximately 20% of all the healthcare-associated infections as a result of the surgical sites. And in Kenya, the rates reported are 7%, 30.8%, and 66%, respectively, from Aga Khan Hospital, Kenyatta Hospital, and Kakamega County General Hospital, in that order. And PA, the Pseudomonas aeruginosa, accounts the highest percentage among the isolates which are isolated. Now, P. aeruginosa, as you know, is a very notable pathogen which is associated with this infection because of its versatility and resistance and the ability to cause the severe infections. And now the emergence of the MDR strains also complicates the treatment, and therefore understanding the antimicrobial resistance profiles is profound, it's important. And the study which we did here aimed at investigating both the phenotypic resistant patterns and genotypic diversity of the organism. So what is the problem? The problem is that we have a problem in addressing the challenge of PA-associated surgical site infections because of this organism which has increased resistance to antibiotics. This organism, which is very resilient, it has the ability to acquire resistance and virulence at very high rate, and this threatens the human health. And therefore, because we do not have limited information, we have very limited information in our on hospital transmission, and also the genomic data is not enough. The phenotypic data is not enough. And this creates a critical gap in surveillance and also characterization of the organism isolated from this. This is very important, or it's required because we needed to have develop a tailored treatment strategy and also infection control measures to be improved and improve the patient outcomes. Now, our study focused on exploring the genomics of the multi-drug resistant PA, and our first step was to determine the prevalence of the organism PA, and then evaluate the antibiotic resistant profiles, and then assess the genotypic diversity in the organism, and finally determine the physiological traits of the organism. So it was very important because some recent studies in Kenya have already identified the globally distributed strains of PA, which are high risk, and also local clones, which exhibit an enhanced pathogenicity and antibiotic resistance. So us getting more insights or doing a comprehensive characterization was actually to provide the deeper insights about the organism, its behavior, and how we can be able to inform effective treatment protocols and infection control strategies. So it was done in Kakamega County, specifically in Kakamega County General Hospital, but the specimens came from some various parts of the county. Now, our methods in the laboratory included the recovery, identification, and used a panel of antibiotics of around 7, which are discs which are commonly used in the hospital for the AST. Other laboratory methods included getting the genomic DNA, extracting the DNA, doing some sequencing under MISEC Illumina platform, and identifying the antimicrobial resistance and virulence genes in these organisms using the respective identifiers, and finally blasting the chromosomal sequences so that you can be able to search for the internal fragments, and also, internal fragments for alignments in these isolates. So what were our findings? When you look at these two represented graphs here, we see that there is a trend of, about the prevalence that 8.9% accounted for PA amongst other, as compared to other organisms which were isolated, the gram-negative rods, the Staph aureus, the Klebsiella, and so on. And therefore, when you look at the second graph, is showing the frequency of the individual microbes according to gender. It's actually not significant because of the p-value of 0.942. It is actually distributed in both sexes. Other results we got about antibiotic resistance is actually the interesting trend of some of the antibiotics, how they actually responded, how the organisms responded to these antibiotics. We find that meropenem is one which was found fit best for the treatment of these surgical site infections by PA, while amikacin counted for almost 0% sensitivity, meaning the resistance was so high as compared to other antibiotics. And a combination of antibiotics actually showed fairly distribution of, accounting for a better option of treating rather than using a single antibiotic, like when you use that piperacillin and the tazobactam and the piperacillin alone, there is a change. Now, when you look at the MDR isolates of the PA, there is also a concerning trend of higher resistance across many of the isolates, and several isolates, for example, the ones with the blue bars, the ID 004, ID 004, 006, 13, and so on, they exhibit a very high resistant level, indicating that these isolates are actually largely unaffected by the antibiotics tested, suggesting they are multi-drug isolates. When you look at the genotypic diversity, very quickly, we see that there is the distribution of the virulence factors. We look at the coding sequences in the chromosome, the coding sequences in the plasmid, and the GI, the genomic islands, because this is where we find the virulence genes and those resistant genes. So we find that there is a distribution of virulence factors in different genomic islands among the organisms. And there is also presence of genes in both core chromosomes and plasmids. And the biggest genome, of course, length also accounts for some 9.7%, while the least is 3.8. Now, plasmids, you see, they were observed only in isolates ID08 and ID15, with the respective numbers of genes as shown, 6 and as shown, 48 and 3. So when you look at the next presentation, is about drug resistance gene, where we find a panel, a number of antibiotics which were exposed to these isolates, and we find that the PA isolate exhibits a very broad and varied resistant profiles across multiple antibiotic classes, and there is high level of resistance. We can see resistant to aminoglycosides, resistant to carbapenem, resistant to fluoroquinones, and so on. And to note here is the presence of resistant genes against disinfectants and antiseptics, which further underscores the resilience of the isolates, making them formidable adversaries in both clinical and environmental settings. Now, the other presentation is about mutational factors. We can realize that mutations are found distributed, widespread distribution of this, and it is consistent, present across all the isolates. We have some mutations found in isolates, which showed in something like fluoroquinolones. You see they have zeros, all of them, but we have one, ID17, which is showing, actually pointing to a more complex, probably potentially environment-specific mechanism of this organism, or to bring resistance to fluoroquinolone. And it is contrasting with the more uniform mutation patterns observed in other genes. Look at these DRG mechanisms. We find that among the antibiotic-resistant genes, the major group was the broad-range efflux pump as a mechanism. The distribution of the virulence genes, we find that virulent genes were only present in plasmids of sequence type 16, that is ID008, and also just to show that the highest, the one which showed highest was ID014, and the smallest number is found in some isolates, which did not even account for any sequence hit or other any, they're like isolates that were not actually known, okay? So we move forward and see how we got the genetic relatedness of the PA isolates, and we find there are two clusters that we found. Now this phylogenetic analysis was very consistent with the sequence typing, where the two clusters of the sequence types were observed. Now, two of them, that is ID020, ID006, are actually unknown because they never showed any sequence hit. What does this mean? Could be it's due to incomplete data or the presence of novel or uncommon sequence types that were not identified in the typing process. This is the physiological traits of the PA isolates from the surgical sites infections. They have various activities which bring about the virulence of the organism and resilience of the organism and the persistence of the organism in the surgical sites infections, including motility, including caseins activity, that is the proteolytic activity, including the exoproteins produced by these organisms, including the production of the pigments of the organism, therefore showing the resilience of the organism. Now, I've discussed a number of this, but I can say that the quorum sensing system was conserved across all the study isolates, highlighting that kind of the evolutionary advantage in promoting the PA survival and adaptation in wood environments. Now, to conclude is just to say that the prevalence is the PA, which is the highest among the isolates, and on multi-drug resistance, we have, resistant rates, resistant rates to common antibiotics, and genotypic diversity, there is also significant genetic variability, and there is novel sequence types which were identified in this testing. The physiological traits, also there's a diverse traits, for example, the ones I have just mentioned. Now, we recommend improvement in infection control protocols. We can also try to integrate our genomic data, which we have, into hospital infection control and the national AMR dashboards. And we can also be able to prioritize culture, which we have been singing now and then about the antimicrobial resistances, practice routine monitoring and adjustment to prescribing practices. We also need to implement some routine genomic sequencing, if possible, and molecular typing to monitor strains and resistance patterns, which these organisms show that they are, again, they have some evolutionary kind of a trend. Now, we should also look at the targeted therapeutic interventions, that is by developing new antibiotics, antivirulence agents, and even personalized treatment strategies, which we should be able to do. I want to thank my team. This is my research team. Some come from the University of Cambridge. We have some from the University of Pretoria, and members of Kakamega County General Hospital, and some from Masinde Muliro University of Science and Technology. Thank you. God bless.
Let's appreciate her once more for that fantastic presentation. Aeruginosa in surgical site infections in Kakamega County. I come from somewhere close to Kakamega County, so I'm just trying to bring this home once more by saying that this can happen to anyone. Antimicrobial resistance is a problem that's facing the whole world, and it can come down to an individual level. So when we see these presentations being made here, it can be you, it can be me, it can be a family member, because it's not just numbers. These patients that have these resistant bacteria, it's people that we know, it's people that we interact with. So again, just driving the message home, let's act now. And by saying act now, who are we telling to act now? Is it the president? Okay, it can't be the president. Is it the CS Health? Yes, but it's also you and me, right? It's also the person sitting next to you. So again, let's continue spreading awareness, and not just this week. Let's not forget about AMR after the 24th, right? Even after this week, let's continue sensitizing everyone around us to use antimicrobials well. So now I'd like to request the presenters to come to the front for our Q&A session. I hope you've written all your questions down. We've had presentations from the 4 presenters, from machine learning to analysis of somatic cell counts in milk. So I hope you've put down your questions, and now we'll be passing around the microphones for anyone who has a question. So we can take questions. There's microphones passing around, so just raise your hand.
I have 2 questions. The first one is the presenter on mastitis. I want him to make a comment on the resistance to Klebsiella pneumoniae in Kenya in general, because I did visit some farms in Bahati, Nakuru, and they did indicate that Klebsiella pneumoniae mastitis, they were having no drug to treat. So during your study, what did you find out, and how does it compare with the other isolates in Kenya? Then the second one is the presenter on Pseudomonas aeruginosa. I would like her to comment on the type of genes she isolated. Have they been isolated anywhere in Kenya? And comment on the epidemiology as compared to the where else they have been isolated in the world. And of my concern, did you detect any resistance, novel resistance that has not been isolated elsewhere in your genomics? Thank you.
Thank you for those very good presentations. My question also goes to the presenter on the mastitis. Given the very high level of resistance that is seen with mastitis. Do you think vaccination does hold any promise, and what challenges are there with vaccine development, if any?
Yes, thank you. My name is Neto from Kisumu. My question goes to the AI presentation, where they had used quite a number of models. And you talked of logistic regression and XGBoost being better than logistic regression, but what didn't come out clearly was the sensitivity and specificity for logistic regression was not, I didn't see it being shown there. And also there's another study which was done on pharmacovigilance, which I attended a conference, which again was comparing the two, but again it showed logistic regression was better than XGBoost. So I don't know if it's the type of data that determines which model to use, or you can just expand on that. Thank you.
Thank you. My name is Dinda Kusu from Japaigo. First, I really want to thank the presenters. Excellent quality presentations. And my question goes to Sarah Kidiki for the excellent presentation, but also more from, translation of what we are seeing from the study to actual practice. The recommendations are on point, but as I listen to these good presentations, I'm wondering, besides this conference, which then is happening in November of 2025, how are we on an ongoing basis using this excellent work within the facilities that we are working, the universities, to actually advance containment of AMR, to weave them, like whether it's in existing committees to drive antimicrobial stewardship practices, to drive the infection prevention control agenda. The question may be directed to Sarah, but I want to even hear from the presenters or anybody how we are really translating this good studies, good work to actual practice to contain AMR in our facilities and communities. Thank you.
Thank you for the good presentations. First, my name is Ogalo. I work with the patient organization, that is the Confraternity of Patients Kenya. And I'm very impressed that you kept on talking about the recipient of care. And my question goes to the AI comrade, the machine learning comrade. Currently, the Kenyan system, the healthcare system has adopted an EMR system. I was just wondering, is this possible that you are able to incorporate or rather integrate those findings, particularly in clients who have been able to identify cultures being done, and so that upon probably another incidence of treatment, they are able to have their data on drug resistance already enlisted during the treatment, so that it becomes one way of also combating AMR. Then our colleague, comrade who did the PA, probably I didn't pick this. Was there any sampling on the surfaces of the hospital, or it was just the patients? Then number two, on the same question is, are there factors that are considered beyond the patient factor during the research, so that probably will consider the environment around? Number two, drug factor on the part of efficacy. Thank you.
Good morning, everyone. Thank you very much for those very excellent presentations. Mine is more of a comment and also just one question. It was very interesting to see the opportunity for us to use AI and machine learning to be able to predict possibility of resistance. We think there's also an opportunity for us to use our own Kenyan data from the central data warehouse, because we've seen from machine learning, the more data you have, the better the models. So we are looking at opportunities for us to maybe partner and work together to see whether these models can assist. I think we also learned quite a number of things, especially on my studies. I was asking Romana, what's the difference between cow level and quarter level? So those are new knowledge that I've been able to learn, and thank you for that presentation. Lastly, I saw from the presentation from Dr. Kindiki on the difference between SSI rates in Aga Khan, KNH, and I think KCGH, and there was quite a big difference between the SSI rates. Maybe you can just mention which study that was, and probably the publication, so that we can follow up. Thank you.
Okay, over to the presenters.
I hope I'm audible enough. About Kleb pneumoniae, we had only one isolate, and it was susceptible to all the panel that we tested against. It was Kleb oxytocin. Most probably because the area is well structured, so most of these pathogens, maybe they're well managed. About vaccination, personally, I advocate for biosecurity, and vaccination is one of them. The use of vaccination in our setting to manage mastitis, I'm not sure if we are at that level yet, but if we can get to that level, it will be good. Thank you.
Thank you for the concerns and the questions. I think I will start by talking about the translation of our research to the community down. And I want to say that from where I sit, because I teach, we have a number of ways of disseminating this to the community around us, including the county and even beyond the county, where we have been able to try our best with the limited resources we have to hold a number of outreaches with the students, the medical students who are doing medical laboratory sciences and public health and all that as a school. And we've been able to disseminate this information by way of awareness through outreaches down to where we hold things like AMR games, we hold some talks with the students and the community just to bring research home. Only that probably the resources are not enough, but we try to do so that we can be able to disseminate. Other things is a matter of joining hands with the county teams, the hospital, Kakamega County General Hospital, where we have collaborations of the researchers and the researchers also in the hospital, where we look at the MTCs, we look at the CASIC, we look at all those groups. We work together and try to mitigate some of these things. Like we can do some research and call upon for some activities like preparation of those agents for disinfection and all that can be used in the hospital. So there is a number of things that are going on in terms of translating the research down to the community. Now, somebody asked about where the research was done. So we have looked at some of the researches done in KNH by Mozilla and the group. It's quite a big number of people who have worked on this, where they collected samples from various counties in this country, including Kericho, Kilifi, some parts of Mombasa, and all that, and worked from KNH and came up with some results where we find some genotypes which were recovered from all these counties, some of these counties which were recovered, and they were actually, some of them were unknown, others were local, others were like, they are transported from far or beyond our boundaries to our country, and they showed that kind of a resistance, and they showed some kind of evolution. And just to join with that, even in our research, we found some 2 isolates which looked like they are Kenyan isolates. That's why we have recommendations for future research through probably cohort studies and even longitudinal studies, so that we can be able to establish how these organisms are active or their evolution, they are actually coming about in Kenya. We have looked even beyond the boundaries, like in Nigeria. I may not remember very well the work done in Nigeria. Another one, there was also some published work from Webuye. I think when we look at these published works in Google Scholar and somewhere, you'll get some of these. And Dinda from Aga Khan also came up with that percentage of 7% in Aga Khan Hospital. It was a number of people who did that research, and they came up with that kind of an infection surgical site rate in our country. Other areas includes other parts of the world, including the European countries, and the resistance we have found. I'm saying it is telling with some of these researches done in these hospitals and in these outside Kenya researches. I think we can get more from the published work. Thank you so much.
Thank you so much as well. I think the first question was on the specificity and sensitivity for the log regression. So when we conducted the study, remember that log regression was our baseline model. And so to understand its performance, we took AUC as the main measure because it accounts for model performance across all classification thresholds. And when it performed lower than the XGBoost, we then advanced with the XGBoost because we realized it was a better functioning model. I would like to request that if the person who asked the question is still would still like to see it, I'm happy to do it as soon as I sit down on R and share that with you. You also asked why I think the question was why we decided, why we mentioned that XGBoost was better, or why we settled on that. Was that the question? Yeah, so AMR patterns are very complex. And so if you take a log regression, which mostly works best for linear relationships, then you will miss out on the nuances that exist within AMR data. XGBoost, on the other hand, takes into accountability the non-linearity of the data that we're using. So it gives us a better overview of the nuances that exist within the data itself, and it accounts for that. It also accounts better for missingness of the data. And one of the limitations with this data was missingness. And even though we imputed some of the data, we had to actually, lose some of it that was over 40% missing. So we had to exclude that. The other question was on, I think it was the general question on translation to practice. I think for us it will take some time because we need to conduct extensive external validation before we can come up with something that is evidence-based and supported by the current evidence that we have in Kenya, which is why I really welcome the, the potential partnership that Dr. proposed. So that's what we're looking forward to. That's what we want to do. And we want to produce a dashboard that also now ties to the other question about the EMR. We want to come up with a dashboard that is easily integrated into the current systems that we have. And so that it's not additional work for the clinician. It's just, just a small portion that they fill out, the key information, and then it gives them, a possible risk prediction that can then also be, it just gives you an idea. And then, once the full results are reached, you can then make your final decision. But it gives you an idea of what's the likely situation based on the research. Thank you.
Probably there were no questions addressed to me from the questions. So may I take this opportunity to introduce myself? I forgot to do that at the beginning of my presentation. So yeah, my name is Teresia Nyaga. I work at the National Microbiology Reference Lab. I have been working with Fleming Fund, which is coming to an end. And there I do a lot of microbiology analysis, a lot of PCR work, and data analysis. Thank you so much.
Okay, thank you to all the presenters. Any final question? I'll give a chance to one last question. Then there's a question online. Maybe you can project that. So I'll read it. It's addressed to Dr. Sarah. Pseudomonas has a lot of intrinsic resistance, which limits treatment options. What are your thoughts on management of these pathogens from your experience? As you think about that, we can have a question from the audience. Yes.
Yeah, my name is Tracy Murea from React Africa. Mine is actually not a question, but more of a comment to sort of encourage the presenters and to tie it with Dr. Tanui and Dr. Kusu's sort of comments. A lot of work is going on at the global level scene in terms of AMR, and especially to influence policy. As we speak right now, there's sort of final consultations on establishing what the AMR political declaration that was agreed upon last year by our leaders in New York to put in place a governing system for known as the Independent Panel for Evidence on AMR, right? Today is actually the last day of consultations. Why am I bringing that up? I do, I work in the policy space, but my background is pharmacy. We are advocating and we are lobbying and we have made sure that that particular final draft or founding document, as they're calling it, takes into consideration that the expert committee of that panel, yeah, is mainly comprised also of researchers and scientists from the low and middle income countries, from the developing countries, and that they should not only look at well-published data, right? I think Gitao started off by saying there's lack of data on AMR in our setting, right? And we've seen that even in yesterday's high-level presentations. We have encouraged that panel and the expert committee also considers the data and information that's not necessarily published in peer-reviewed. So for instance, what comes out from such poster presentations or from such forums, my encouragement is put it out there, because it means once this panel is in place, then we'll start getting data that is context-based to our regions that then informs what we're finding at WHO as priority pathogens and the antibiotic matching to actually inform production of the novel antibiotics that Dr. Sarah talked about. So mine is more to encourage, and not only the presenters, but the whole audience. Please give us that data, give us that information, let us see it out there, because it will come to actually then inform what is context-based and what can guide us empirically, even if in the space of shrinking global health financing, meaning that we might not honestly be able to get targeted therapy to the extent that we want in the short term that we want. We might have to think about empirical therapy for a longer time. So that means we need better evidence as is being brought out from here. Thank you.
Okay, thank you, Dr. Tracy, for those remarks. So the final question, oh, sorry.
Thank you for the online question. I think the online question was talking about the resilience of this organism and what we can do, or what I think. Now I can talk from probably a personal background, or I mean a personal perspective, and not actually written down anywhere. I'm not referring anywhere. So this organism, we have seen that the PA is so resilient. It has so many mechanisms of virulence. That means it can adapt very fast in the environment. It is an organism that is so persistent because of those traits. It has some traits of that kind of adaptability to the environment, and those virulence factors that lead it to be all that stubborn. Now, from where I stand, I can only continue to adopt what the comment we have gotten from the experts, another expert there, that we strengthen the IPC protocols wherever we are, and try to make awareness. Other things we can talk about, trying to look as a country, or maybe the policymakers, we try to look for ways of creating even new antibiotics. This requires a lot of research about it, because we need some maybe preclinical trials, we need clinical trials to find out whether they are safe, whether they are, and all that. So we need interventions like even looking for newer antibiotics about these organisms that are evolving every time. And also, we can target on some agents which can be able to be antivirulence, targeting something specific, particular, some certain genes, not necessarily what has been there. Because of this evolving, we can get the virulence factors and we target. So we look for antivirulence agents and try to do some personalized treatment strategies. Thank you.
Let's appreciate all our presenters for today, for this morning. You can have your seats. And before I sit down, I also have two things to mention. Number one, the QR code for registration, which is right outside here. It's working now. So in case you haven't registered for today, please make sure during tea time you pass by there, scan the QR code and register. The registration is for every day, so So the homework is look up if that antibiotic is on the the aware classification. If it's access or watch or reserve on the WHO classification. And report back to your neighbor after the break. Okay, thank you.
So please give a round of applause to Michelle, my co-chair. And also give yourselves another big round of applause for being a wonderful audience. The online team, you can also share some reactions, maybe some claps to show that you're part of us. So we've come to the end of our session, and we have about 15 minutes for the poster session. So I will request all of us to just walk to the posters. Do we have the poster presenters in the room? Maybe you can raise your hand, a poster presenter. So please just walk to your posters, stand close to them in case we'll have any questions from the audience coming to view. Then minutes after that, we can proceed for the break. Thank you all.
Address AMR. So we still have a long gap in terms of capacity building, and I'll call again the stakeholders and partners here, kindly support capacity building in a way that we can be able to build champions who can be able to go, even if they just send a message via peer learning, telling their colleagues that this is what we can do, this is how we can be able to prevent AMR together. Then the other one is leveraging on existing structures. I think as an organization, we've been able to work closely with NASIC, and that has really built us. I remember I could be able to jot down. We have the Nooksound food for thought. Set of presentations. We'll be currently pursuing an MSc at the Cambridge Graduate School, learning medical the potential of bacteriophage to combat drug-resistant infections. The potential of bacteriophage to combat drug-resistant infections. He founded the Phage Hunters Training and Research Program to build East Africa's first course based on graduate research experiences, in brackets cures, providing students with
it 100 times, and it's not quite scary. But if you compare that to a plane crash, because if a plane crash were to happen right now, one plane that I think the largest carries about 400, one plane that I think the largest carries about 410 people, the Boeing, the headlines from CNN to Citizen and whatever. Those numbers, if we were to compare MR numbers of plane crash, we will be losing, we've been having 9 planes crashing every day for a year for us to get to 1.27 million deaths. So, hope that brings teaching. That we, granting on the science courses, particularly focus on the medical science because I'm a medical scientist. Majority of the graduates end up in medical science schools. So these are people who go to school this problem, but they're not actually addressing it. Why? Because maybe they didn't have the mentorship or the guidance to pursue it as a research interest. And that's what we are trying to address by saying we need to build that interest within the young scientists or our undergraduates before they even get into graduate school. So this is how phage works. You can see it attaches to bacteria. Our chicken will die from infections. love what Professor Maponga said, if you know it, he said, our education system is flawed because it doesn't prepare our young people to address problems in the community. Because I need to go to school when I know AMR is a problem that is affecting my mom at home who has cuckoo. So I need to find a problem of how I can prevent this spread of AMR in my local community. That way we have young people who are driven to solving this problem. And the problem of having scientists moving away from the field will be, I hope, STEM. I'd like to acknowledge all these people because most of the work we've done, we've not been able to do it alone. We have been able to do this work because of collaboration and working with different people across the board. The theme for this year is Act Now, protect our present, secure our future. And I'd urge you, if you're here, a lecturer, a researcher, any key stakeholder, please join us on this journey of making FEJ a research priority in the country. And hopefully, I'm hoping in the next 5 years, we can be able to celebrate and say some of the stages that have been isolated by our students are actually used in therapy to stem the problem. We are currently enrolling for a training for our Journal Club Forward for 2026. So please feel free to recommend this to your students or anyone in your networks. Thank you so much.
Thank you very much, Duke of Kakamega Shire, for the insights on young FEJ scientists. I'll take this opportunity to welcome Cynthia, again Duke, and Ndege. We want to have a brief Q&A session before we proceed to the next set of 3 presentations. So Cynthia, Ndege, and Ellis, and welcome to the podium. We'll take questions in sets of 3. So kindly raise your hand, and my colleague Mkapa will reach out to you with the microphone. Any question?
Maybe I can start.
Okay, sure. Thanks.
Thank you so much, presenters, for that insightful session. I think my question may be tied, so you can decide. I believe we're doing well in engaging the youth, and Cynthia and Dege have been champions of the youth from day one. But just tying into the work that the youth are doing in research and innovation, even beyond pages, looking at data utilization, AI, and all that, do you feel that the engagement is adequate, or if it's not, where can we plug in in terms of the national program for AMR containment? Where can it be plugged more? And then maybe directed to Sir Kakamega. In relation to pages, I saw your partners and I saw Kipper there, but I didn't see Kenny. And I'm not sure whether the page bank is actually sitting with them. So I wanted to know what is the link you have with the research that Kenny is doing with pages.
Good afternoon, my name is Felista from the Act Africa. The question goes to Ngere. I know you mentioned that you've not yet measured the outcome of your program, but what are the plans and how do you intend to measure the outcome and success of that biosecurity study? Thank you.
Thank you. My name is John from ICANN. I have a question for Cynthia. I think involving the youth is a smart move. However, how do you ensure reports lead to action in terms of decision making or shaping AMR interventions in the community? Thank you. Thank you. Let's have those 3 questions responded to before we proceed to the next 3. Thank you. Over to speakers.
Okay, thank you. I don't know if I should start. Okay, I'll go ahead and start. First of all, I'll respond to Katha's question in terms of if the engagement with young people adequate enough, especially in terms of research and maybe in other initiatives. I would say no, it's not enough, but I would also acknowledge that there's some existing platforms that engage young people, as much as it's not enough for overall youth engagement because of some limited opportunities that currently exist in the country. So one of the ways that we can maybe ensure adequate engagement within the existing national structure is in terms of having internship opportunity for young people, especially what the FED team is doing, just recruiting young scientists in activities that they're doing. So this one I'll also call for other partners who have different projects, if they can have an open opportunity for young people to apply for internships in terms of the activities that they are doing. Even at the national platform, I would say at the national government, there's that the government, it's called Public Service Internship Program, where young people are being placed in different areas to work or to participate. And to the national level, the NASIC, I would say yes, there's an opportunity for young people, especially in the Youth in Action initiative, where we engage young people across the country on matter related to AMR. And to respond to Dr. Joseph's question on how to ensure that young people's suggestions or ideas are included in the table, most of the time it has not been an easy journey just to even get that platform to showcase your idea. But I will comment I'll just talk on behalf of the NASIC that personally I'm part of as the secretariat. For me, we see that most of the things that we propose at the NASIC level have been included. So what we do at the national level is just to give opportunity for young people, even at the county level, to coordinate and work with the CASIC, that is the County Antimicrobial Stewardship Agency. That's why we say that we are kind of linking the young people at the county with the county MR focal points, so that they also get opportunity to participate in the roundtable discussions on matters related to AMR at the county level. So I'll say it is still a journey, but at least we are making some progress. Thank you.
Thank you. So the first question on are we doing enough in terms of engagement? I'd say we're doing work, but we could do better. And why I say this is because I feel the MR problem has been glorified among scientists a lot. So we love talking about it in conferences and among scientists, and get excited because my results have found a resistant pathogen, so I'll have something interesting to report. But is it getting to the common mwananchi? I look at what happened with HIV when I was growing up. Everybody knew about HIV. I mean, it was even in the curriculum within primary school. We had a class on HIV. Why don't we have a class on AMR? I mean, it should be there, because it starts with making sure the young people or the children even understand. When I go to a doctor and he gives me an antibiotic, I need to finish my prescription. I don't need to share my drug with my brother, so that when you have an infection at home, you know, kids are very good. They even tell you, "Chichali niambia." So if you're a parent and you see a child has a cold and you're giving, I think a cold is a bad example, but has an infection, and you're giving them an antibiotic, if they're siblings and you try and use the same prescription for the other one, the kid will be like, no, these ones are mine. So I feel we need to do more work in terms of making sure that the conversation of the AMR and raising awareness reaches to the communities. I am a child of a farmer. We rear chicken at home, and I have seen firsthand how very unregulated drugs are used in livestock production. Why are we not controlling that? What are the vets doing? Why is it that it's very easy for me to approach any agrovet and ask for an antibiotic to treat my chicken, and it's easy for me to get it. And even the one who is giving me won't tell me how to administer it. I'll go and do it on my own. So I'd say, yes, there's work happening, but we can do better. On the question of partners and community, I think I should have mentioned the acknowledgements that I put here, most of those are our friends. Our key partner is TUC. So we have not directly engaged with Cambridge, but we have worked with some of the scientists there as our advisors. For example, Dr. Msila has been very instrumental in advising us on how best to proceed moving forward. So just to say, we are very open to working with everyone. On the phage bank conversation, at the moment, I know every institution that is doing phage is keeping their own phages. I don't know if we have a national phage bank. So I'm also in the unknown. So our work is just trying to tell the country and everybody involved that we need to make phage a priority. This phage bank was needed 10 years ago. Why don't we have it right now? So if you're here and you can help us speed up this conversation, make sure that we can get these phages to patients who need them, we are happy to work together. Thank you.
Thank you. First, I'd like to respond to Dr. Felissa's question about how do we measure the outcome of the farmer field engagement. And as I mentioned earlier, one of the assignments that we were giving the youths to do was to visit the nearest farm. Sorry. One of the things that we were doing, we were telling them, go to the nearest farm, observe this and that. Are they doing that or not really? So they were able to give us feedback, but that was informal. Now, when we're going to do the farmer visit, we're going to do it in two phases. One, they're going to do a pre-visit. They're going to observe and they're going to record down. We have what we call farmer commitment card and a toolkit where they're going to respond the key aspect of security that they implemented. So, are they having a fence at the poultry house? If yes, no, that is what is going to guide us on the intervention that we're going to recommend to them. Once we recommend the interventions, we're going to go back after 14 days. That is 2 weeks, and we're going to check the commitment card. They committed to make sure that the poultry house is cleaned maybe twice a week or maybe even once a day. Are they doing that? Then we're going to get between pre and post. If there is improvement, then we're going to say our farmer field visit was success and we were able to make impact. Then on adequate youth engagement, I'd like to commend the NASIC for making sure that every year we do have the youth event subcommittee. Because in countries like Nigeria, I had a conversation with a colleague, I think last week, and he was saying, Kenya, you're doing amazing work. Youth are doing amazing work in Kenya because we are doing activities during 1 week that is coordinated by the government. For them, they don't even have a contact of the person coordinating the NASIC in their country. But here in Kenya, we have a privilege to having the youth event subcommittee, and that is one of the avenues that youth have really been able to implement some of the activities, and they've been able to build a track record that can be a reference point on their capacity to be able to do activities. However, there still exist some gaps, especially in terms of policy development and engagement. I think last year, as Africa Youth Amr Alliance Task Force, we were able to do a position statement for the UN General Assembly. And I think that was at the regional level. At the national level also, I think we also need to include the youth in terms of policy development so that they can be able to have the experience as early as possible. So in the future, they can be able to be very experts in terms of policy development in containment of the AMR. And On the youth, young people ideas acceptance, I think so far I've seen you need to build trust amongst the stakeholders so that you can be able to, your ideas can be able to be accepted. Once you have had that trust, I think it can be easier for your ideas to be accepted and be implemented as part of the intervention strategies across the globe. Thank you very much.
Thank you very much. I think in the interest of time, I know there are a couple of questions. We still have 3 more presentations. In the interest of time, I'll kindly request that we bring this half session to a close. Thank you very much, our presenters, and a round of applause to them. There are 2 comments, or 1 comment there on the screen from John. Hi to everyone, thanks for the presentations. Our next set of presentations, our presentation will come from Odiwar Byron and the Phoenix community-based organization in Kisii County. He will share with us insights on a multi-year One Health grassroots AMR program, combining mentorship, cycling, campaigns, school clubs, and community engagement. Byron Odiwar is the Secretary General and a driving force behind the Phoenix community-based organization in Kisii, Kenya. He's a passionate advocate for youth-led development. He spearheads the organization's flagship AMR cause. Under his leadership, Phoenix CBO has mobilized hundreds of youths, healthcare workers, and community members in innovative Amr Awareness campaigns, establishing him as a key figure in advancing the One Health agenda at the grassroots level. Welcome, Byron.
Thank you so much. As it has been said, my name is Odiwar Byron, Secretary General of Phoenix Community-Based Organization that is based in Kisii, specifically Kisii Central. Today I want to just talk about the AMR course that we are doing as a CBO, that is driving community change through the youth-led initiatives. On the background of Phoenix CBO, I would wish to say that this CBO was founded back in the year 2020 after we recognized that empowered youths can do better. Empowered youths can have a best place in the community and help us move the agenda that we have as a community. The mission was to champion global and sustainable peace through youth empowerment. We had a vision that is to empower community that serves as a catalyst for social transformation. The core values that we were having is the value of irreplaceable dignity of persons under the common umbrella of humanity, irrespective of the race, religion, political persuasion, economic status, gender, and ability. Okay. On the issue of antimicrobial resistance, we are talking about the global and the local threat. We realized that AMR is a global and local threat as well, and this has caused millions of deaths, and we also, it has also been a global health threat that is requiring an urgent action. And that is why the youths for Phoenix CBO are coming together to help the community be aware of what is happening. The mission for the AMR course is to combat AMR in Kisii County and beyond through awareness, education, and advocacy. Since these are the limits that as youths who are there, we are capable of. The approach, we are using a year-round multi-stakeholder collaboration, partnering for awareness, education, advocacy, engagements, culminating in WOW and leveraging One Health approach. Now, as you can see from the picture, that is just one of the examples of the pictures that we were doing a cycling, and we had the common agenda that we had that day was to tell the community what is basically AMR.
And that is why we had reflectors that are printed, and we had some field information that the community were getting. Objectives of this was, overall goal for this was to promote responsible antimicrobial use and proper hygiene through community awareness, education, and behavioral change in the communication strategy for the communities. Key objectives, number one was to educate students, youth, and community members on AMR. Number two, we have facilitate accessibility of the community level of national development of AMR IEC materials. We also talked about engaging healthcare workers as antimicrobial stewardship mentors. These are the people whom, if we have on board, the youths are able to be mentored and in the correct way on handling the AMR issue. We also foster establishment of sustainable AMR awareness that is structured in schools and community. We realize that when we start from a lower level, then we come to upper levels, a student or a pupil will grow knowing what is AMR and how it affects and how to go about it. And the core that we are now championing is antimicrobial resistance course. We achieved whatever we've achieved through some activities under the AMR.
can still create impact and reduce the progression of this AMR. So I'd like to acknowledge my fellow MUSTAC members, Masinde Muliro, especially the Dean School of Medicine, the IPNET for the continuous support, AMREF, the NASIC, yeah, and Kakamega County. Thank you.
Thanks, Calvin, for highlighting once again, how youth can get involved in the fight against AMR. I think this session has really brought out to us the role that the youth can play in creating awareness on AMR. Some places I believe, most of us would not be so willing to go, or even if we went, we may not have, the lingo, the language to be able to communicate. To the border people or fellow youth outside there. It's now, I think, about 1:10, and I don't know whether I'll be right to say that our lunch duration will be inversely proportional to the number of questions that we ask. Is that a fair assumption? So I'd like to call the presenters to come on stage. So like I've said, the lunch duration is inversely proportional to the number of questions that we ask. Our presenters, kindly find your way to the stage. We will start off with Dr. Kusu. She has some comments on this session, and then 2 more questions, if we're in agreement. After Dr. Kusu, 2 questions, and then we can break for lunch. But if we want more, we can do that math, isn't it? Dr. Kusu.
Thank you, Dr. Collins, dear moderator, for allowing me to just share 2 or so comments linking between this session and the previous session. And it's very clear, and we want to applaud the youth, that really the youth have a critical role to play. They are energetic, they are innovative, and they are on the ground, as we've seen. And one of the questions we've been asking ourselves, even we've spoken with a few of my colleagues, is how can we paint the country, youth and AMR, looking back to 2019. And I think if there is a way we can cascade the Phoenix CBO model, or even the students across the country, that's gonna help us, so that we don't have to keep explaining ourselves when we discuss AMR, which does not have a face like HIV, for example, has. My second comment was on the phages, and I say that with passion. Thank you to the gentleman who discussed phages. But one of the things I would urge NASIC, the One Health AMR team, is if they can spearhead a conversation around phages, bacteriophages. It's more than having a bank of phages. It requires, there are regulatory aspects, there are policy dimensions. We will not just take phages and use them in our, in our patients, but we really recognize the work and the many, people involved in the different aspects of research. So that's to NASIC. Then the last comment quickly. Gere highlighted something, and it's a cross-cutting something, around funding. And he said the youth are not, they may not be committal, they may be fluid, and the issue of partnerships are to mobilize resources. I'm making this comment because it's appearing in many of the slides around challenges, whether it's the youth or other sessions. But then how can we think outside the box in terms of the partnership?
work being done. Okay, so we are coming to the end of our session. Let's clap for our presenters. They've done a good job. Okay. I think I play a lot and work less. We can clap for Diana. Thank you, Diana. Now, I'm impressed by the number of questions we had in this session. Now I have a reward for you, which is a game. Who's ready to play as we go? No one. No one is ready to play a game? Huh? You want to go home? I'll let you go home. Yes, no? I'll let you guys go home. No. Anyone else has a question to make? None. Tucheze a game basi. Come. So the reward, do we have sweets in the table? Do you still have sweets? Mmemaliza. I have a reward. Tea is ready. So we can take tea. I have two or three, 4 questions. Please project. So this game is something you'll see visually, then you say what it is. Gen Zs. Sijui mnyamaze. Mtanyamaza. Susan says she's a Gen Z and she's going to be silent. Okay. I'm sure those ones on TikTok, they've seen this before. So what statement is this? It's a statement. Ukisema jibu unaenda chai. No? Doing better? Yes, you're doing well. Bado? Life after death, correct. Sweetie, unaeza enda chai. Tea is ready. It's life after death. Aya, this one? You're almost there. What did you say, Nancy? Growing economy. Very good. Hapana. Mko karibu hapo. Hiyo arrow iko wapi? Where is the arrow? Someone said the answer. Yes, center of gravity. Aya. Another one? That one is easy. Well done. Aya, watu hesabu? Well done. That table is very alert. Tendeni chai. Hiyo ni Gen Z. Wamesema nyinyi ni Gen Z. Yes? Okay, tumepata. Zimeisha? One more. Hapana. There is a head. Headquarter. Right. Clap for yourselves. Clap for yourselves as we close. All right, well done, and thank you so much for your participation. We are closing the meeting for today. We see you tomorrow. It would be so un-African of me not to have someone close the meeting with a prayer. A volunteer? A volunteer to help us close the meeting with a prayer?
So we thank the Lord for the good day that He has given us. So we are going to pray. I think I will request we can stand. We pray together. So let us pray. Father, in the name of Jesus, we want to thank you because of this time. Lord, you have been so gracious to us. Right from morning up to this time, we want to thank you because of what you have enabled us to do. And Father, we just give you praise for the far that you have taken us. Lord, even as we come to the end of this day, We thank you and give you all the glory. Even as we go wherever we are sleeping, I pray that you shall protect us, and even as we come tomorrow so that you can be able to finish, we pray that your grace and your protection shall be together with us. We even want to appreciate there are some families maybe at home for some of us. We pray that you keep them and protect them. We give you praise and worship your name, for it is in the name of Jesus we pray and believe.
Amen.
Amen.
Thank you so much. All right, so we've come to the end. Tea is ready outside. We meet tomorrow at 8:30. We start at 9 a.m., but please be here early. We start at 9. Asante.